Today we are going to have 95th lecture of Tamil Nadu Science Forum, jointly organized with the Anna Centenary Library. A small introduction about Tamil Nadu Science Forum. Tamil Nadu Science Forum established, it's an NGO established somewhere in 1980s for popularization of science, and it is spread across Tamil Nadu. All districts have its structure is there, and mainly into popularization of science and education and women empowerment. Like that, 6 verticals are there. Okay, and this lecture series started somewhere in 2018, and it was started offline, and when the COVID came, it was in the hybrid mode. And now also it is hybrid mode. You can watch all the 94 series of lectures in YouTube. It is there. Okay. So with this, I go to today's. It is my pleasure to introduce our esteemed speaker for today's lecture, Dr. H. Shakila. Is renowned expert in the field of immunology with over 2 decades of experience in research and academia. As a retired professor from the Department of Molecular Microbiology at Madurai Kamaraj University, Dr. SHAKILA has made significant contribution to the development of DNA vaccines for infectious diseases like tuberculosis and HIV. Today, Dr. SHAKILA will be speaking on a topic that has recently garnered immense attention in the scientific community. That is, peripheral immunotolerance can save our lives. This topic is particularly relevant this year, as the 2025 Nobel Prize in Physiology or Medicine was awarded to Mary E. Brunkow, Fred Ramsdell, and Shimon Sakaguchi for their groundbreaking discoveries concerning peripheral immune tolerance. In her talk, Dr. Shakila will highlight the importance of peripheral immune tolerance in preventing the immune system from harming the body. She will also discuss how these discoveries have paved the way for the development of medical treatments for cancer, autoimmune diseases, and may lead to more successful transplantations. I request Dr. Shakila to take the stage and share her valuable insights on this critical topic. Please welcome Dr. Shakila.
Thank you, sir. Very good morning to one and all. I'm so happy to see all the enthusiasts. I should say enthusiasts because I find a population of students and also all retired people and going to retire, common public, everybody is there. That shows how much of enthusiasm you have for science. So here, I'll not be talking much or deliberating a big talk about immunotolerance, which itself is a special topic in immunology. You should know that what is happening in our system. So you all know that we have a very well developed immune system, and that is what is protecting us throughout our lifetime from the day we are born. From the very day we are born up to the last life when we leave. So till then, our immune system is going to play a very important role. And not only that, you will be amazed to hear that even before the birth, when the fetus is developing in the mother's womb, there also immune system plays a very important role because it has to protect the baby and from various infections, because we are exposed to the various infections. So, and also many other reactions which are happening inside the mother's womb. So that also may cause some danger to the baby. So from the day the baby is developing and from the last life of our life till then, our immune system plays a very important role. So that is a small pretext I wanted to give about the immune system. And you should be very happy that all of us who are living a very happy and hale and healthy life should be thankful to our immune system, because without which you will, it's really what to say, it's really difficult to lead a normal life. So let me start with these three scientists. You all know that for the past 2 weeks, we have been talking about Nobel Prize in various subjects. But we are going to discuss today about the Nobel Prize, which has been given for physiology of medicine. So every time they give, or every year they give in this field, making sure that some contributions should be given to the human being. So it has to, at least some clinical trials should have taken place where something positive has come up for helping the human being. So that's why the Nobel Prize is given. And what is the reason for giving the Nobel Prize for these three people? So what are all the contributions they have made? That's where I'm going to discuss about this one important topic, which you will find very difficult to pronounce also. Peripheral immunotolerance. Why immune tolerance should happen in our body, and what is it? It is playing a very important role and contributing for our health, and how these three investigators or scientists played a very important role in this particular contribution. So let me go one by one previously. So the three scientists, after after all, this is the first, I should say, only very minimal women scientists have been rewarded or awarded with Nobel Prize. So we have to actually give special credit to this particular lady, who is Mary Brunkow. Now, she actually, even though she was from Princeton University, she settled herself in the Institute of Systems Biology, which has nothing to do with, you all know that system biology is related to computational biology. So she is now settled here, even though she has done her biology part previously. And with this help, she could decipher so many things. So that is her contribution. I will tell one by one, like how these scientists played a very important role in identifying the role of this peripheral immunotolerance. The next person is Fred Ramsdell. Again, he is from USA, University of California, and now he is settled in a industry which is called Sonomo Biotherapeutics. So you see how again there is a transfer of scientists, no, not involving themselves only in the research institutes. They go into the industries also for developing various products. So that is Fred Ramsdell. And the third one, who is the senior most of all the among the three, is Shimon Sakaguchi. He is from Japan. So he got his PhD from Kyoto University. You all know about that. And he is a distinguished professor at the Immunology Frontier Research Center, and he is an immunologist also. So what is this? These three individuals or these three scientists identified. Actually, it didn't take them 1 or 2 years, almost more than 2 decades, particularly Sakaguchi. He worked for more than 2 decades for identifying only one type of cell, which I will tell you later on. And then these two scientists, they worked also for deciphering what could be the gene which is really important for one type of autoimmune condition. Now, why autoimmune diseases are formed? Now, this is where your immunotolerance is very, very important. So that's where I'm also going to going to give some inference about what is autoimmunity, how autoimmune diseases are seen in many individuals, why some are healthy and some are not healthy, getting this in autoimmune diseases, what is happening in their body. So that's where all the answers for this peripheral immunotolerance will be pictured out. And that's why these three people were given or awarded Nobel Prize. So to start with, I will just give a basic information about immune system. You all know that. From top to bottom, we have so many of these nodes. You can see that nodes are there. They are all lymph nodes. So lymph nodes you see from top to bottom, from our head till our legs. You find throughout our body, we find these lymph nodes. What are those? They are your secondary lymphoid organs. And what is the primary lymphoid organ? Then we have to mention about thymus and bone marrow. So these are the two important organs which we have in our system, and they play a predominant role in generating different types of cells of immune system, and also playing a defensive role whenever a foreign invader comes and sits inside our system. So our immune system, pictorially, I may sound very like it's a very basic thing, but it's very, very complex, because you have primary lymphoid organs in the form of thymus and bone marrow, and you have secondary lymphoid organs, which are nothing but the lymph nodes, which are distributed throughout your body. And there is one major secondary lymphoid organ, which is spleen. You all know that it is, again, it's a most important organ of immune system. And apart from that, we have some patches and patches of cells, which are also distributed throughout your gut, throughout your intestines. So they are all referred as Peyer's patches. So these are all the organs which contribute for the immune system. So we have organs, we have everything. So what is the role of these immune system? So they are, next slide please. So they are supposed to do three major functions. You all know that the first and the foremost function is they have to defend ourselves from the invaders, the foreign invaders. Who are those? The organisms, microbes. You're all well aware that throughout us, like throughout this atmosphere, we are surrounded by millions and millions of microorganisms. So every time these microorganisms, when they enter our body protects us from this microorganism. So how does it happen? It is all because of your immune system, which plays a very important role in the defensive role. So the major function is it protects from disease causing invaders. And the second important function is it also does a duty of scavengers. You all know that so many debris get settled down in our system. So who will clean your immune system? Or who will clean not only immune system, the other regions of your body? Again, the cells of the immune system, they act as scavengers, like how corporation people will come and clean. So similarly, without this cleaning process, your body will not be healthy. So you need to have somebody to do this scavenging activity. So there also your immune system plays a very important role. And the third important role is your immune system recognizes and removes the abnormal cells. So who are the abnormal cells? Naturally, you would have heard, you have been hearing also about tumor cells and cancer cells. So they are all abnormal cells. So when a cell is not normal, when it is sitting inside your system, it has to be removed, and it has to be recognized and removed immediately. So who does that? Again, your immune system does this. So now you understand what are all the major functions of immune system. Apart from its defensive role, it helps in cleansing activity, it helps in recognizing and removing the unwanted cells also. So these are all the major functions. So since I already discussed with you about the organs of the immune system, you are also aware of the cells of the immune system, nothing but our own WBCs. So you know that blood system is made up of red blood cells and WBCs, which is white blood cells. So all the white blood cells come under the cells of the immune system. So they play a major role in doing this defensive role. So you have different types of white blood cells. So here we are going to talk only about these cells which are going to contribute for the immune system. So what are those? Next slide, please. So if you want to see, these are all the cells of the immune system. So you will be amazed to see that some 10 to 12 cells are involved in our immune system, which contributes for this defensive role. So starting from T cells, B cells, and all these cells. I'm not going to talk about all these cells, but these are all the cellular makeup of the immune system, and they are in general are referred as white blood cells. So in today's talk, we are going to give much importance to this particular cell, which is called T cell. So I'll just go in detail about this cell, because it's not that the other cells are not doing any role. Each one of them has got their own role to play a defensive mechanism in helping us to remain healthy. But now the Nobel Prize has gone to these three scientists because of identification of one group of T cells which is going to contribute for getting treated for autoimmune conditions. So that is the most important thing which these three individuals have highlighted, and that is why T cells are very, very important in this context. Next slide, please. So I'm just showing a blow up figure of the immune system. This is how it looks like any other system, like what we have: circulatory system, nervous system, then you have a gastrointestinal system. So similarly, we have one system called immune system, which is where all the factors, all the cells of the immune system, they all network with each other and play a very important role in not allowing the foreign invader, that means the microbes which is there in the atmosphere. When it enters into the body, it is protected by these cells of the immune system and these organs of the immune system. So next slide. So I'm just giving few examples to show how each of these cells, they contribute in killing the microbes. So there is a photograph showing that there is one cell called neutrophil. You all know that I showed in it. This is also one type of WBC. And this cell, you can see, it can extend its arm. See, naturally, if you see a neutrophil, it will be spherical in structure. But when it senses something is coming near, it will go immediately and grab that microbe and eat it. So that is the major function of neutrophil. So neutrophil, it swallows and gulps or eats the microbe, and that is why it is referred as a phagocyte. So like neutrophils, you have one more cell which is called as macrophages. So they are very powerful. See, macrophage also, you can see it is just developing some dendrites, some arms like structures have developed, and they go and grab the microbes and they eat it away. So they are referred as phagocytes. So we have powerful phagocytes which can recognize who are these invaders, and accordingly they will take action to eat them, swallow them, and digest and process and present it to the next group of cells, which I'll tell you in detail. So this is just a reference to show you how powerful our cells of immune system are. And then thanks to B cells, now you would have heard that during COVID-19 also, you all know that we all develop good antibody response. So who is that antibody? Who is the supplier of antibody? Again, the B cells. So B cells, they differentiate themselves as plasma cells and they produce antibodies. So these antibodies only will go and attack all the antigens, and that is how we are all taken care when some microbe enter into our system. So you see, in the beginning itself, you find all these cells coming into the picture and trying to get rid of these pathogens as early as possible. Next slide, please. Now, our area of interest today for the topic is the T cells. So T cells, again, why T cells? Because T refers to the group of cells which matures and differentiates from thymus. So since it is developing from the thymus, so it is referred as T cells, and they are the major cells again which contribute for a very good defensive role. So you have two important types of cells. One is called helper cell, T helper cell, which is in the center. The other one is T cytotoxic cell. So till 25 years back, people were aware of only of these two cells. They didn't have the knowledge of about the other third type of cell, which is called regulatory T cell, which is what we are going to discuss further. So T cells, basically, the helper cells, they are enormous in numbers. The name itself indicates they do the helping. So they always come into the picture. Whenever a microbe enters, this T helper cell will come immediately and immediately signal the other cells to get ready and alarm the immune system. See, you all know that when somebody rings our calling bell, immediately we get to know that somebody is outside, and so we should get ready to open the door. So similarly, this fellow T helper cell, it just makes an alarming signal to the immune system and tells, say, look, some virus has entered. Look, some bacteria has entered. And there also, it discriminates between what type of bacteria has entered, whether it is a gram-positive bacteria or a gram-negative bacteria. So all these informations, these T helper cells will have, and tries to communicate to the other cells so that all the cells will be getting ready to get rid of this pathogen as early as possible. So that's the role of T helper cells, and that is why they are referred as helper cells. And we do have among all these T cells, the highest number of T cell population we have in the form of helper cells only. So you find around 60 to 70 percentage contribution is coming from T helper cells. Now, why less contribution from cytotoxic T cell, the next group of cell? Because the name itself indicates they are toxic in nature. They destroy the pathogen. There is no nothing about that. They do their job. But at the same time, the cytotoxic T cell activity can be damaging to our own tissue also. So that is why they are referred as cytotoxic. So this toxicity can not only damage the pathogen which has entered, but it can also damage our own tissue and it can cause some injury. That is why by nature, we are having less number of these cells to have in check. So always among these two groups, helper cells will always be in higher proportion and cytotoxic T cells will be in a lower proportion. And they will come for action only during viral infection and only during tumor activation. So tumor elimination. Because only to these conditions, cytotoxic T cells will come, but for rest of the pathogens, your helper cells and other cells like macrophages, dendritic cells or B cells, they all will come into the picture to save the host or the human being from the pathogen. Now, I'm going to talk about this third variety of T cell, which is called T regulatory T cells or T reg. You can, in short, you can call them as Treg cells or T reg cells. Now, the name itself indicates that they are regulatory. That means they play a very important role for balancing your system. Because you all know that anything with too much activity is not good for the system. So somebody should come and suppress. Because sometimes that's why we are all healthy. That's why we don't have autoimmune diseases. The others have. That's because in people who develop autoimmune diseases, what happens is their immune system is in hyperactive mode. They don't have that much of Treg cells. And that is why that hyperactive mode of the cells are controlled by nature by the Treg cells. So T regulatory cells, they play a very important role in suppressing your immune response. Why should there be a suppression? Because always active, overactive always is not good for the system, for the body. So something by nature should be there to bring down this overactiveness, and that is contributed by the Treg cells. Next slide. So that's how all these cells come into the picture. But naturally, if you see, when a pathogen enters, the first cell which comes and grabs the pathogen is the dendritic cells and the macrophages, which I already told you, they are phagocytes. So they will first find out where is that pathogen sitting. They will go all the way, grab that fellow, swallow, eat it, and break them into pieces and then present it to T cells. So this is how the networking between the dendritic cells, macrophages, and T cells happen. See here, one thing I would like to tell, among all the cells of the immune system, everything can handle the pathogen by themselves. But T cells cannot handle by themselves. They have to depend on antigen presentation. That means somebody should hand over the antigen to the T cell, then only this fellow will become active. Till then, T cells will remain in an inactive state. Whereas all the other groups of cells, they can handle their antigens or the foreign pathogens by themselves. Whereas T cells require the help from the antigen presenting cells or the phagocytes. So that's why macrophages, dendritic cells, these cells come into the picture. Next slide. And they present the antigens. For example, here you can see a virus cell is infected by a virus, and you know virus infection, virus immediately goes inside the cell. It doesn't remain outside the cell. So it enters into the cell. So when it enters into the cell, it will immediately, the antigen will be expressed by the infected cell. So any infected cell, viral infected cell, they will try to express an antigen, the viral antigen, and that will be recognized, sensed, and signaled by the phagocytes, and they will come and try to capture that antigen and then present it to their T cells. So that's how T cells immediately come into the picture there. And I already told you, for viral infection and tumor, your cytotoxic T cells, they come into the picture and they try to kill the infected cells. So they not only kill the pathogen, but they also kill the cell which is infected. So that's the thing about cytotoxic T cells. Next slide, please. So this is again a photography to show how T cells can attack a cancer cell. So cancer cell, again, they will be producing or they will be expressing some cancer antigens on their surface, which will be recognized by our cells. And so T cells go there, grab that cell, and they will try to kill that cancer cell or the abnormal cell. So this is how each of these cells, they communicate with each other and convey the signal and tell them that somebody has entered or some abnormality is happening. So all the cells should come there and kill the tumor cell. So that's what is happening here. So as soon as, see, why I told you cytotoxic T cells are lesser in number, because I already told you that this damaging effect what the cytotoxic T cells cause not only damages the pathogen, but sometimes it can cause some tissue damage or host damaging also can happen. That is why you find a lesser number of cytotoxic T cells. Next slide, please. Now, our topic of interest today is T regulatory cell or the regulatory T cells. Now, they come in the picture when they sense that too much of hyperactiveness is happening in the immune system. So, when hyperactive nature of the immune system happens, these cells will assemble and they will try to suppress. So, they bring about what is referred as immunosuppression. That is very, very important, because every time if your immune system is active, it is not good. It should be active when in the presence of a pathogen. But when the pathogen is cleared, it should remain in a naive state. That means it should remain in an inactive stage. But in some individuals, what happens is this activeness goes above and above, and so you find some hyperactive reaction, and because of that only they develop autoimmune condition. So to bring down that type of hyperactive reaction only, your T regulatory cells are going to play a very important role. Next slide. So here comes the tolerating mechanism. Why should we tolerate? Who should tolerate this? So this is referred as immunological tolerance. That means our immune system should recognize only the foreign antigen. That means the antigen which is coming from the outside. Whereas the antigens which are produced inside your body should not be recognized by your immune system. It should tolerate that antigen. So that type of tolerance is called as immunological tolerance. That means you tolerate your self antigen and your immune system should react only to the non-self antigens or the foreign antigens. So this mechanism is called as immunological tolerance, and this is the one which is divided into central tolerance and peripheral. Why central tolerance means by nature in thymus and in bone marrow itself, the T cells which have recognized our self antigens, they don't react. They are stopped. They are suppressed there itself. So that's called central tolerance. But sometimes these cells may escape from the thymus and bone marrow and they reach other peripheral tissues, and at that stage you find these TREG cells coming into actions. So T regulatory cells, they are present everywhere, and they are the ones which are responsible for this tolerating capacity, tolerance, which is very much needed so that they just bring down or suppress our too reactive immune system so that we remain healthy. Next slide, please. So this is how the harmful T cells, like why I should say harmful, because T cells, after their action is over, they should calm down. They should just come down. They shouldn't overreact. But in some individuals, these T cells start overreacting, and so they are referred as harmful T cells. So they should be cleared from your system. And who does that? Again, In the thymus itself, we have some negative selection is there, some other mechanisms are there, T regulatory cells are there. They play a very important role in bringing down this hypersensitive T cells. And next slide. And next, in the peripheral tolerance also, next slide, we have this important role of T regulatory cells. They contribute for this immense mechanism of suppressing our immune system. See, I will tell you why our immune system should be suppressed and in what are all the conditions should we suppress the immune system. Because we always talk about your immune system should be good, very active and very active mode, but not in hyperactive mode. So that is the difference between why we should suppress and why we should not suppress. So here, next slide, please. Now, this is where I would like to highlight these three important things. Now, what will happen if this peripheral tolerance is not happening? See, in normal healthy individuals, in all our body, peripheral tolerance is happening because we have T regulatory cells and they always suppress our immune system when in not use, when it is not in need. At that time, the peripheral tolerance should be checked. So who is doing that? Again, we have various factors. Among them, most important factor is T regulatory cells. Whereas, if this mechanism fails, what happens? You will get autoimmune attack. And this autoimmune attack, I can give various examples starting from type 1 diabetes, rheumatoid arthritis, and other autoimmune conditions like systemic lupus erythematosus, and many other examples are there. Till now, they have identified more than 100 varieties of different types of autoimmune diseases. So for all these things, for all these autoimmune diseases, even though genetic factors are also contributing for autoimmune conditions, even though environmental factors have also contributed for autoimmune condition, now these three people have identified saying that immune dysregulation that plays a very important role in the contribution of autoimmune diseases. So we have all the three factors which contribute for autoimmune conditions, out of which your immunity, that is immune response, reacting too much. There is no one to suppress that immune reaction. Because of that, people have got autoimmune condition. And that is why if peripheral tolerance is lost, we develop autoimmune conditions. So these three people were able to prove that. Next slide, please. So this is one famous photography you find in any of the immunology books. You see here the dragon is eating its own tail. So similarly, our immune system is damaging our own tissue. So this is what is happening in the case of autoimmunity. Why it happens? It is all because of the tolerance, that immunological tolerance or the peripheral tolerance or the central tolerance, whatever it is, this tolerance is lost in some of these individuals. Next slide, please. So good example I would like to just cite here is type 1 diabetes mellitus. You all know that pancreas is a very important organ where in the pancreas, the Langerhans cells, the pancreatic beta cell is very important for secreting insulin and hormone, which is what is very important for our glucose cycle and everything. But here, what happens is in type 1 diabetes mellitus, our own T cells go and block the pancreatic B cells, beta cells, and also the antibodies which we produce, they go and block it because of this hyperactive reaction. And because of that blocking nature, enough insulin is not secreted by the pancreas. So here, the culprit is our own T cells and B cells. They are the culprit here in the case of type 1 diabetes mellitus. Next slide, please. In the case of rheumatoid arthritis, again, rheumatoid arthritis is a condition where the synovial membrane, you all know that that is a membrane which is lined throughout our joints, our elbows, knees, everywhere, the synovial membrane is there. Now, this synovial membrane tends to secrete some antigens, which is our self antigens, for which we should not react. But in these patients, what happens, since they don't have this tolerating capacity, their own T cells and their own B cells and macrophages, dendritic cells, all the cells, they go start accumulating in the synovial membrane, and they block in all the joints, and that is why you find the swelling in the joints in the rheumatoid arthritic patients. So here again the culprit is our own cells are causing this particular damage. Why here it is happening? Because here nobody is there to suppress this hyperactive reaction. So the immunosuppressor is not there, and that is why these people are affected by rheumatoid arthritis. And so how do we treat these conditions? So all these years, we have been treating autoimmune patients with immunosuppressant drugs. They are themselves very highly toxic, and they themselves have so many other side effects. And we are giving such toxic chemicals to treat these or autoimmune conditions. No other go, because they have to live, and they have to improve their livelihood. So these people are treated with such powerful immunosuppressive drugs. And this one important drug, you must be knowing, cyclosporin and methotrexate. Actually, it's a cancer. It's a cancer-causing drug. So no other go. So they are giving that drug for treating autoimmune condition. And tacrolimus and prednisolone is a steroid. Again, steroids, they are also harmful. And tacrolimus and cyclosporin is a fungal metabolite. We have taken the drugs from the fungal species, and that has been used as a drug for treating these patients. But they also have their own side effects. Next slide, please. So you find, I just listed here all the side effects caused by these immunosuppressants. So now we need not worry, because these three scientists actually have shown the way wherein we can use these TREG cells, T regulatory cells, which are already available in your system. But in those patients, they are not working. So maybe we can treat these individuals instead of the drugs. We can treat them with T-reg cells of population. So that's what they have highlighted in this peripheral tolerance. And most importantly, that is how they have indicated the role of T-reg cells. Next slide, please. So here, what did Sakaguchi did, or what Mary and Frederick did is, this Shimon Sakaguchi actually for more than 2 decades, he has worked on the autoimmune conditions, studying the autoimmune diseases, and he was the one sole responsible for identifying this new class of T cells, namely T regulatory cells. So special credit has to be given to these Japanese scientists who has identified this group of cells, and he clearly pictured out that it is these cells which play a very important role in immunosuppression, and it is these cells which are in need for treating these autoimmune conditions. And what is the contribution of Brunkow and Frederick? Actually, they also worked with autoimmune conditions, many autoimmune diseases, but prolonged work they did on one disease called, it's not a disease, it is a disorder called IPEX syndrome. Now, IPEX syndrome is a syndrome which is referred as immune dysregulation, polyendocrinopathy, enteropathy, and it is X-linked IPEX disorder. So people who are suffering from such disorder, they could not survive for a very long period, and they had very many problems to undergo. We could not treat those individuals also with other immunosuppressants which was given for other autoimmune conditions. So many things were happening. So they were doing some animal trials, preclinical trials, and even some clinical trials they have worked, and they were the ones who have identified that one particular gene which is called FOXP3. Now, that is the one which is very much responsible as the transcription factor for the development of T regulatory cell. So, the Japanese scientists identified the cell, but these two scientists identified the factor, the gene which is responsible for the development of this T regulatory cells, and that's how all the three contributed for this one important mechanism which is called as peripheral tolerance. Next slide, please. So how do these FOXP3 regulatory T cells work? Now, what is the mechanism behind them? How can they bring about immunosuppression? You will be amazed to see, like, whether is there such things are happening in our system. We are totally unaware. After these three people only identified all the mechanisms of the T regulatory cells, even the immunologists now they are fully aware. Okay, so many things are brought about by one single group of cells which are called as T regulatory cells, and they are playing this multiple role. So what are those things which the T regulatory cells contribute? Next slide, please. So T regulatory cells, actually how they bring about the immunosuppression is, they can actually deprive the cytokines. You know that cytokines are the signaling molecules, the mediators, which helps in communicating one cell to the another cell. So if your immune system is too active, hyperactive, this T-reg cell will come there and it will try to deprive all the cytokines. So all the cytokines will come down so that the active T cells will now become inactive. So this is one type how or one way through which the Treg cells bring about immunosuppression. Then another important methodology is cytolysis. Cytolysis means Treg cells has the capacity to kill any type of cell which are overactive. So how do they do that? Again, Treg cells, they produce large amount of some enzymes called granzymes and perforins. They are powerful enzymes. So these enzymes will be produced by the Treg cells and they start bombarding the other immune active cells which are there nearby. Thereby, they bring about immunosuppression. So this is second mechanism. And the third mechanism is T regulatory cells produces another group of cytokines which are called as anti-inflammatory cytokines. That means you have a group of cytokines which are called as pro-inflammatory cytokines which bring about inflammation, whereas Treg cells will lower down the inflammation. It will suppress the inflammation, and so it produces cytokines which brings about, which changes that entire environment of inflammation into anti-inflammations. Thereby, it controls the inflammation and suppresses your immune system. So this is third mechanism. And next slide is, yeah, next slide. Next slide is, here also there is another powerful mechanism which happens by Treg cell. What it does is, it starts digesting or eating, I should say consuming lot of IL-2. See, IL-2, interleukin 2 is an important cytokine which is needed for the development, growth and differentiation of other T cells. So what this fellow does, Treg cell, it eats away or it swallows all the IL-2 which is needed for the other T cells. Thereby, it starves the T cells not to become active. So they are starved to death. They even die. The other T cells die because they are deprived of IL-2, which is a very important cytokine which is needed for the T cells growth. So you see how intelligent, I would say, this T regulatory cell is so clever to bring about this immunosuppression by various mechanisms. No, it produces one cytokine and it tries to eat away some other cytokine, and it can kill the other active T cells. And it not only the T cells, it can also kill macrophages, dendritic cells. It can make them inactive. So all the active cells are now being converted as inactive cells thanks to T regulatory cells. So because of one group of cells which are referred as T regulatory cells, you find these multiple mechanisms which are happening. Thereby, your entire immune system gets suppressed. So that's how your immunity gets suppressed when not in need. So your immune response should come up, should be active only when in need. Otherwise, it should come to a suppressive mode. So who brings that? It is all due to our T regulatory cells. Next slide, please. So this is again an important mechanism. I was really wondering, like I too started I wanted to work with, my students are also there. We were thinking of working with T regulatory cells in role for tuberculosis control, HIV and TB HIV. All those things we were thinking, but we couldn't work out. But it's amazing to see that T regulatory cells can produce some vesiculated structures which are called extracellular vesicles. So what are these vesicles? No, they are vesicles which contain rich enzymes, rich molecules, which will go, travel, transported, get transported anywhere and try to target whatever has to be targeted. So you find so amazing about this T regulatory cell. This T regulatory cell can produce this type of these many different types of mechanisms wherein it brings about immunotolerance. So one important mechanism is developing extracellular vesicles or EVs, and thereby it tries to suppress our immune system, and it can induce apoptosis. So you all know that apoptosis is a cell death. So who brings about that cell death? Naturally, any cell, whatever is born, it's programmed. It is programmed to die by some days, some weeks, some hours. It is all depending on their life cycle, they will die. But some of these hyperactive cells, they remain alive. So who brings about the death of those cells? Our T-Rex cells. So T-Rex cells, they have that opportunity of even bringing death to the cells which are not in need now. So that is what T-Regs are doing. So this is called apoptosis, which is a major mechanism which helps in clearing the cells if their duty is over. Next slide, please. So now I'm coming to the clinical side of you. So why am I talking only about T-Rex cells? What is their role at the clinical perspective? Is it going to be really useful for human beings? Yes, because I already told you this cell is going to contribute for autoimmune conditions, diseases, for controlling autoimmune diseases. It is going to play a very important role because clinically, these scientists, they have done many clinical experiments and they have shown that all the trials, at least up to clinical trials, whatever they have done till now, they have proved and showed that T-Regs are going to play a very important role for controlling the autoimmune conditions. So that is very important. I'll just show few examples. Next slide, please. So autoimmunity, now people are thinking about cell therapy. So you just infuse T-Reg cells to the patient who are in need of T-Regs. So who are the patients who are in need of T-Regs? Patients who develop autoimmune conditions. So autoimmune diseases, you all know that it is because of various factors. Some may be due to genetics, some may be environmental factor, but now people have shown that it is mostly due to immune factor. So if you want to bring down or suppress that hyperactive nature of the autoimmune condition, you can add T-Rex cells, like how people supply blood to other individuals. So similarly, T-Rex populations can be given as a therapeutic purposes also. So that's what is being attempted for autoimmune diseases. Next slide, please. And for allergic condition. Now, why are we allergic to some food? You all know that some people are allergic to milk, eggs, fish, and many shellfishes, even peanuts. Peanuts. Some people are allergic to peanuts. Why are we reacting too much? That's because in those individuals, those food which are taken into our gut or into the stomach and then goes into the intestine, that food will be considered as an allergen or it will be considered as an antigen. And to that antigen, our immune system will start reacting, and because of that reaction only you develop allergic reaction. So now, in order to control that type of allergic reaction also, T-Rex can come handy. So they are going to be really helpful for controlling the allergic conditions. Next slide, please. And gut immunity, I should talk about gut immunity because you all know that our gut is made up of millions of microbiota. We have millions and millions of good, harmless, beneficial microbes sitting from our oral cavity till our gut cavity. So from top to bottom, we are made up of these microbes only. So if you disturb the gut microbes, that's where so much of intolerating capacity mechanism happens. Bowel syndromes, irritable bowel syndromes, and diarrhea, all these things happen because of the disturbance what we cause for the gut. So usually the gut microbes should be tolerated by our immune system. It should not react. They are also microbes only, but they are harmless, they are beneficial. So our immune system should tolerate, should show that immunological tolerance against that gut microbes. Then only we will remain healthy. But there are some people who also develop some intolerance, and because of that, their own immune system start attacking the gut microbes and disturbing the gut microbes. Again, that will lead into various other conditions. So here also T-regs can play a very important role. Next slide, please. And tissue repair. Do you know that for many repairing of your tissue, because we get damaged due to trauma, due to injury, or due to some problem which is happening on your skin or muscle, heart tissues, lung, everywhere some damage will be happening now and then. So for repairing such cells also, for repairing such tissue also, now these people have identified that Treg cells are going to play a very important role. So all these days I was telling you that Treg cells are immunosuppressors, but they can also help in repairing your tissue. That's really amazing again. So that is because Treg cells produces one important component called amphiregulin. So this component plays a very important role in repairing your tissue. So whatever damage has happened, that damage is rectified by T regulatory cells. So that is again an important aspect which we should think of. Next slide, please. And coming to repairment. So not only various tissues like placenta damage, skin, skeletal muscle, brain, wherever tissue repair has to happen, there they have shown that T regulatory cells are doing wonderful job in repairing all these tissues. Next slide, please. Now, this is very important system because you all know that we have a blood-brain barrier in the central nervous system. Nothing can cross. So blood-brain barrier, actually central nervous system is a privileged site. So even your immune cells cannot travel to central nervous system. So that's a privileged site. Like that, in our system, we have various privileged sites like, do you know, any of you, do you know, have any idea? What are all the privileged sites where your immune cells cannot enter? Like brain, we have others. No. Cornea, yes, cornea, that too aqueous humor of cornea, and your lens of the eye, and then uterus, testis. These are the regions where your immune system cannot go. They cannot cross. Your immune cells cannot cross. So they are all referred as privileged sites, even central nervous system. So even in that privileged sites, they have identified T regulatory cells. How did it come? So by birth itself, T regulatory cells, the other cells which does not have that tendency to move, but T regulatory cells are there. And they say that they contribute so much for regulating even the central nervous system. Can you imagine? How come a regulatory cell which is belonging to the immune system is communicating some signal which is belonging to the central nervous system? Now that's where they say that there is a strong network between the central nervous system and the immune system through the T regulatory cells. So T regulatory cells, they play a very important role in conveying the signals from everywhere to the brain also. So that's what they say. And not only that, T regulatory cells, they play a very important role in neuronal proliferation, and they bring down the inflammation of the astrocytes. So astrocytes are the cells which you find in the brain. So there the inflammation should not happen. So T regulatory cells, they suppress that inflammation in the brain also, and they play a very important role in controlling one type of other groups of cells called microglial cells. Now microglial cells are, again, they are the cells of the immune system which are like macrophage in nature. So they shouldn't be overactive. If it is overactive, some brain damage will be happening. So T regulatory cells which are present in the central nervous system, they bring down this type of homeostasis also by suppressing the immune response there. So this is the nature of the T regulatory cells in central nervous system. Next slide, please. So we have a strong gut-brain axis. I do not know whether you believe it or not, but scientifically it is proved that whatever you eat is also going to be reflect in your brain. So the gut-brain axis, that's why they say that you don't disturb the gut microbiota. The gut microbiota, which is there in your system, should be preserved well, and they are the ones which are contributing for the good signals which is getting transmitted to the brain, and from brain it comes to the gut. So that gut brain axis, to maintain this also you need T regulatory cells. Next slide please. And Tregs play a very important role in liver, in inhibiting the fibrosis in the liver. Next slide. Next slide. And the most important topic, why all this time I was talking that only Tregs will be useful for autoimmune diseases. No, T regulatory cells are also going to be useful during transplantation. Because you all know that hundreds and hundreds of individuals are waiting for various transplantations in the hospital. So when you go for transplantation, what happens is the donor, whatever he or she donates, the recipient, when they accept, their immune system will start overacting. So because of that hyperactive nature of the immune system, the organ would be rejected very easily. So to avoid that, you can use T regulatory cells hereafter. So hereafter, all these days they were using again for transplantation also, immunosuppressant drugs were used for suppressing the immune system. But now we are having these natural T regulatory cells which are already present in us. So they can be infused to these individuals who are going to undergo transplantation so that they don't react too much and they do not show this rejection. So whatever organ has been transplanted, it should get transplanted successfully. Then only that patient will survive. So for transplantation also, they say that Treg cells are very, very important. Next slide, please. Next slide. This is just to show. And another important area where Treg cells can really be useful is pregnancy. Because you all know that many pregnant ladies, they have the spontaneous abortions and miscarriages and all these things are happening. Now here, again, the most important culprit is again our own hyperactive immune system only. So our immune cells, because I already told you, uterus is a privileged site. This fellow should not go there. But sometimes during this types of intolerant capacity, what happens is all the hyperactive T cells and all the antibodies and all the cells of the immune system, they start going there and they start aborting the fetus. So that can also happen during pregnancy. So here also they say that Treg cells can come really in handy for treating these pregnant ladies, because for successful placentation and for suppressing your immune system, you need the help of Treg cells. So for at this stage also, Tregs are going to be really useful. Next slide, please. Next slide. Finally, why I'm talking about cancer cell? Because cancer, actually, your immune system should be active, very active, hyperactive. Then only you can kill the cancer cells. Here you cannot suppress your immune system. So it's like the opposite side. So, but here also the scientists are thinking that since Tregs are having high targeting capacity, they can target nicely. They know whom to target and they can, like a bullet targeting, they can do very effectively. So now scientists have thought whether they can tie a medicine with the Treg cell and then try to target only the cancer cell so that the other normal cell will not be affected. So by this way, you all know that cancer patients, when they are taking treatment, the normal cells are also getting affected. They are undergoing radiation. They are taking chemotherapy. And all the drugs what they take is going to be toxic not only for the abnormal cell but also for the normal cell. So you have to avoid the normal cells getting affected. So for that, what you can do is you can use the Treg cells for targeting only the cancer cell. So this is being attempted. As I told you, these scientists, they have attempted in various clinical trials. Next slide. Next slide. And these are the experiments what Sakaguchi did. And he only developed this CD4 positive, CD25 positive FOXP3 T regulatory cell. So till 25 years back, when 1992, when I registered for PhD, I just had one line of note in the textbook that there could be one group of cells which are called as T regulatory cells. But afterwards, now they've fully shown that, clearly proved that there are some new group of T regulatory cells which are present, and they are going to contribute enormously for the humankind, particularly for treating autoimmune conditions and also other pregnancy, tissue damage, tissue repairing, and also for transplantation. Next slide, please. And this is the gene I was telling you, FOXP3. So Brunkow and Ramsdell, where they curiously they sequenced all the genes which are present in all the chromosomes. You all know that we have 23 pairs of chromosomes, and these individuals only, they identified that this particular gene is present in the X chromosome. So it's X-linked gene, and so in that also, when they isolate one by one, tediously, they picked up all the gene sequence and then they studied. It's not an easy job. So they did it meticulously, and they found that the gene which was responsible for the development of T regulatory cell is none other than FOXP3. So this is a main transcription factor which is very, very important. So people who are having mutation in that gene, or people who don't synthesize, who don't have this transcription factor, they are the ones who don't develop Treg cells, and they are the ones who develop autoimmune conditions. So that's the conclusion they came finally. And so now all of us are working towards this particular gene. Next slide, please. Next slide. Next. But next. Next. So till now I was talking to you only about all the positive side of the Treg cells, but we should be really cautious. Why? Because we are going to suppress the immune system. When you suppress the immune system, what will happen? You will be succumbing to various infections also. Because when an infection comes, who is the protector? Your T cells and B cells only. You cannot suppress them. So during chronic infection stage, we should be really careful, and also we cannot be completely suppressing or impairing our immune response, because that can lead into various ineffective conditions. And another important thing is cancer also. You cannot fully rely on suppressing your immune system, because for attacking a cancer cell, you need a very good effective immune cell only. That fellow only attacks the tumor. So if you suppress the immune system, naturally your tumorogenesis cannot be controlled. So that also should be taken care. So these are all the negative effects. But in spite of all these negative effects, we do have some positive informations about these Treg cells and their activity and their role in controlling various autoimmune disease. Next slide. Thank you so much. Yes.
Thank you, ma'am. Now that Mr. Sivakurman from Anna Library will honor the speaker.
SHAKILA. Thank you. Thank you.
Thank you. Okay. Basis now.
What was it on the Treg cell is so important? How do we increase Treg cells or genesis of Treg cells? Is there a way for it?
Ways, actually, naturally, we should have been born with good number of Treg cells, and by drugs or other things we cannot do. But they say that we do have some factors like good proteins, protein molecules, enzymes, all these are needed. And that is why they are talking about this adoptive cell transfer. So therapeutically from outside, only you can give inside. People who lack. So the most important factor is this gene, FOXP3. So people who have the mutation in that gene, or who lack that gene, they are only they are deficient in developing this particular group of cells. So genetically, if you want to modify, now what they are doing is, they take the cell, Treg cell, from the genetically deficient individual, correct it outside, and then again infuse it through, it's a stem cell therapy. So that attempt also is being made. But otherwise, normally, we should have been, all of us should have been born with good number of Treg cells, at least to bring about this normal activity.
Thank you, madam.
Thank you for the nice session. So I don't know much about biology or biotechnology, but at any point of time in the last one hour, I felt like I don't know anything. I was very much engaged in the conversation. I really love it. Thanks for that. I have two questions. How these three people are connected? Do they know each other?
No. It's all for, see, Sakaguchi, very senior person from the past 22 decades, he have been working with Treg cells only. But he didn't have the clue like why only some people are having Treg and people who have developed autoimmune diseases did not have that much of Treg cell. Whereas these three people, they were also working, but epigenetically they went and they found out, maybe then only they found out the clue here is the gene, which is very, very important. And they went for some animal trial and clinical trial.
You said that now, when you registered your PhD, that time itself the concept of Treg cells were there, but no one knows its existence.
Yes. They've proved, saying what could be its mechanism and how it gets activated.
Okay. So maybe it's a dumb question. Would there be any medicine or something like that could come, or the medicine will be to make FOXP3?
That's genetically you cannot. Only thing is
out of this discovery, it is a procedure.
Cells. Yeah, the cells only should be given as a therapeutic purposes.
Okay. So maybe how long do you think that this will be widespread for people who are really suffering from autoimmune?
No, it's already clinical trials have started and they have shown proof saying that some 3 phase of trials have started. It's only a final phase. Once it started, it will come to the market also.
Maybe by next 3-4 years.
5 to 6 years.
Thanks a lot. Fantastic.
How does the immune system react to the microplastics?
Microplastics.
How does the immune system react? Does it react or does it?
Yeah, it will react because plastics, it will be polymerized in nature, no? So mostly, see, immune system will react only for the protein structures. So that's why they say proteins are highly immunogenic, antigenic in nature when compared to the polysaccharides or the lipids or anything else. So if that polymer contains some little bit of protein, it will react. Otherwise, it will not react. That is why we find it gets settled down and it blocks and do some blocking thing.
Thank you. Microglia and the cells are there. But the 2 cells, immune cells, present in certain cells.
No, only microglial cells are present. But when damage happens to the brain, when the blood-brain barrier is affected, that time this fellow goes inside and they start hyperactive. Yeah. So there on that time only it goes.
Okay, with central nervous system controls the entire. I mean, not only the balance of happens in the brain, it also provides balance for the entire body.
Yes, yeah. So that's why they say you have a axis, no, gut and brain axis. Not only that, brain controls everything. So it has access throughout the system, but here they have shown clearly that even for immunity also, the gut-brain immunity plays a very important role.
Is there a connection between the lymphatic system and this?
Yes. Yeah. Lymphatics. See, lymphatic system is a complex structures made up of many lymph nodes, and your spleen, thymus, and bone. All these are connected, like how blood system, circulatory system is connected by arteries and veins. So we do have a lymphatic system connecting all these nodes and spleen. That's why when we get some infection, we find the enlargement of the nodes, no? So doctor, when you go, he touches here because tonsil is a lymph node. So when it enlarges, that means it shows that you are having some infection. So all your antigen antibody interactions will be happening only in the lymph nodes. So that's the ground where this war will be happening.
So all this communication is through T cells.
T cells. Not only T cells, we have the cytokines are there, they are signaling molecules, just the protein molecules. Then B cells will produce the antibodies. They also go everywhere and they try to capture. So it's a network of these group of cells. They all communicate together and they bring about a good response. And their only main aim of the response is they have to clear that pathogen. So that's a normal thing. But abnormally, what happens is after clearing also, it becomes too much of active. And because of that, it starts damaging our own tissues. So that is when the T-Rex come into the picture.
To be more precise, the suppression happens through CNS or like through the body.
No, suppression is everywhere. Not only, see, T-Rex is present everywhere. It is happening. Wherever the emergency is needed, it goes there and tries to suppress your the other cells.
So microglia only controls CNS. By suppression in CNS happens
by through T-Regs, not microglia is a type of macrophage which is present in the brain.
What my question is like whether brain controls
Suppression
parts of the body like cancer.
Cancer, yeah, signaling, no? See, all these brains actually they have neurotransmitters, neurosignals are there. It conveys the signals throughout the neurons and everything, and from there it travels throughout your body. But the cancer cells are identified by our immune cells because cancer cells will be producing a different type of antigen called tumor antigens. So those antigens will be picked up by our immune cells and they will start attacking the tumor cells.
About for the poor person or the disease. In the maybe you take
Cells from outside and give it as a therapy. Yeah.
you take from person or from another person like we use.
That should be matching again. It should be mostly, yeah.
If we inside from other person, they consider antigen. So
Yeah. So what you say is correct. Actually, like transplantation also, they transplant from only a donor and recipient matching. No, matched. They should be competent enough. So that is why they check for blood grouping and HLA typing also, and all those things they do matching. So after matching only, they try to donate the kidney, liver, even heart, everything. So blood group. So similarly, one thing what they have shown is they have taken the patient's T-reg itself, they have modified that T-reg and then again infused. Another thing is they have taken close matched another individual. From that reg they have taken, again modified and transfused. Yes, that will be there. Again, that will be there because see, anything which comes from outside will always will be considered as a foreign only. So for that, again, that is why for transplantation individuals, what they do is before they get admitted for transplantation, at least for 1 month, they'll be given immunosuppressants. So they'll be suppressed. Everything will come down because they should not react to the donor organ because the donor organ itself will contain some immune cell and the recipient will be receiving. His own tissue also will be containing some immune cells. So there will be a crosstalk between the immune cells of the donor organ and the recipient. So there itself the fight may start. So for that only the immunosuppressants are first given and then they see the matching. Then after best matching organ only is donated and that is being given. And they'll be monitoring the patients with immunosuppressive drugs only. So here only what you can do is instead of those toxic drugs, we can give the strep population also, which will bring down your immune system so that you don't overreact to the organ. Now he is in need of kidney. Without that kidney, he cannot be alive. So to make him alive, we have to infuse him with various Treg cell population and make him.
Monoclonal antibody with these T cells.
The
All
monoclonal antibodies with
T cells. Specific T cell we
get
monoclonal antibody.
For targeting. See, actually monoclonal antibodies only are used for treating tumor. Tumor will have some antigen. To that antigen, that monoclonal antibody will be targeting and that's how they kill They treat the tumor cell. So nowadays, people who have been treated for cancer are being given for monoclonal antibody therapy only. So like that, now they are attempting to attract the TREC population there. So with the help of monoclonal antibodies also, you can attract the TREC population and try to, not for cancer. See, for cancer, actually, your immune system should be at the higher mode, not at the lower mode. So for other conditions, you can bring this attraction of TREC cells into that particular area through monoclonal antibodies. That can be.
Is brain transplantation, how much is successful? Brain transplantation.
It's very difficult. It's not that successful. The most successful transplantation are kidney, liver, of course, skin transplantations are happening everywhere. That's all.
Brain transplantation will make behavioral changes.
Must be there, yeah, because again, brain, you cannot preserve it, no. See, heart transplantation also is nowadays happening. That too, they say within 6 hours the patient die. Within that 6-hour period, the heart has to be transplanted. So only these 3 organs have been successfully transplanted. Brain is very difficult because
Regarding DNA, how far is it gone back? There is
how far
how much it is going to go beyond.
this autoimmune condition, it all depends on where the gene, no? If it is X-linked gene, see, that's what I said, one syndrome, IPEX. If it is X-linked gene, it is mostly actually, so you know that male have XY chromosome and female have XX chromosome. So, so mostly, again, if it is X-linked, that is why most of the autoimmune conditions, it is female. Because of double dose, it is present in the X chromosome. Whereas in male, it is only one dose, only one X is present. So getting autoimmune diseases in males is lesser when compared to females. And again, genetically, it depends on how much of inheritance is happening. It is all like anytime mutation can happen, spontaneously it can happen, anything can happen. So that we cannot say anything now. It depends on how many generation it goes.
Basically, I'm not very good background. First question is, I see whatever you talk about cancers and this kind of things, there are developments that is welcome to the appreciate. But why this is it on both it's
You mean cancer? Cancer. Why? Because polluted. Everywhere pollution. Food is adulterated. Water is adulterated. That is polluted. Air is polluted. No, 20 years back, my grandmother lived so happily up to 96 years of age because she had a healthy food, healthy atmosphere, healthy lifestyle. Lifestyle, again, our lifestyle has changed.
exactly. So probably first thing what I want to see is, for example, it's not for explicitly saying, just what I understand right from. I'm getting up morning every day, 3:30, 3:40 per day, and I'm taking leave and just for drinking and then doing yoga. And then I'm taking protein supplements, ingredients, and then and other things, and sleeping, completing my food by 7 o'clock and sleeping by 9 o'clock. So do this really is the immune system or anything else?
It will, but how is your physical activity?
Yeah, morning I do 1 hour yoga.
Yeah. So how active we are physically, that plays a very important role. And all the food, what we take, that is also very important. No, healthy. I would say whether it is organic or inorganic, whatever. It's a healthy food. No, your gut system. That's why I said your gut microbe. So many millions of microbes are present in our gut. They play a very important role in digestion. You should not be eating or loading them with lot of all unwanted foods. Yeah, for some stage, because that's say actually fasting also is very good because at least that gives some rest for the cells, because every time it keeps on working and working day and night, and at least some time you should give some rest. And what we do is either we give some rest or we fully load. No, you shouldn't be like that. You should do some justice for the gut, wherein we take whatever is, they say usually you should fill your stomach only for 85%, not fully loading. And then you give limit timing and water, of course. Actually, I should say previously we were all having well, we had groundwater, we drank that water, and so we were living healthy. Now water itself we are buying and packaged, we do not know what is added there, and we are drinking that. So everything, environmental factors also contribute a lot for immune system. Yeah.
And the thing is, when we need to fix with our lifestyle and other things,
Mm.
then we are trying to fix that later part
Mm.
to working on the cells. Right? That is well and good. But however, mostly
From adult stage is the stage, no, like all productive adult stages from the teenagers up to, I would say, up to 50, 60 also. After 60 only, because you all know that maybe by, as an immunologist, I would say thymus, which is a primary lymphoid organ, it will start reducing in size when you age. So when I go beyond 60 or 65, the thymus will start shrinking. So when that is shrinking, naturally your T cells also will come down. So when that is coming, your immune response will come down. That is why more elders are prone for more types of infectious condition than the young people. So that is natural thing which is happening. But apart from that, we have other things also. So why don't you boost your responses by taking healthy diet, healthy lifestyle, and physical working, and all those things will help us a lot in boosting our immune system.
thank you. We're going to the next question. From YouTube, nearly 20 people are watching. One question is there. Now, as it is, how do tumors recruit and expand Tregs in the tumor microenvironment?
Yeah. See, actually, in the tumor microenvironment, only the other T cells, like T cytotoxic cell, which is very, very important, that only should come and play a cytotoxic role in killing the tumor. So that is what is happening in many of the individuals who recover from cancer. You see many patients recover from cancer, like colon cancer, breast cancer. Many of them have recovered along with the therapy. So along with the therapy, they start recovering because our own cytotoxic T cells, they come there and they start recognizing only the tumor cells, and they start attacking the tumor cells and kill the T cells. That is one thing, good thing that is happening. If there the T regulatory cells come, they will start immunosuppressing, which is not needed there. So there you need only hyperactive nature of the T, cytotoxic cells and other cells, macrophages, dendritic cells, all these cells also come and they produce powerful enzymes, proteins, and B cells also come to attack the tumor cells by producing antibodies. All these things are happening. When this is happening, that itself will slowly clear the tumor. So at this stage, you don't need the T regulatory cells, because there you need the help of other groups of cells to recover ourselves from the tumor so that it can kill the tumor cells. So tumor Treg cells, if it is recruited, it will be recruited by the tumor cells, because you know that sometimes some phenotypic changing, like they say that it is known as T cell editing can happen. So what this tumor cell does is, instead of bringing the good cells which are going to remove the tumor, it starts bringing the Treg cells which are going to suppress your tumor. So by the not suppressing the immune response. So by this way, tumor cells also can sometimes dictate the immune system. So when they dictate, your entire strategy changes. So that is when the Treg cells come into the microenvironment, tumor microenvironment.
Thank you, ma'am, for your lecture. My question is, you have mentioned that if any other cells are hyperactive, then these red cells will go and suppress them. What if that red cells itself become hyperactive? Is there any separate mechanism to control the red or hyperactive?
Yeah, we have other mechanisms also. No, it is already there. Actually, Immunosuppressors also should be kept in check. If they keep on proliferating, again, it will start keep on suppressing our immune system, which is not good because we are surrounded by millions of outside organisms. So when they enter, the other cells only going to help, not the Treg cells. So in that case, the immunosuppressors should be suppressed. For that only, we have some cytokines are there. They are referred as pro-inflammatory cytokines. So these cytokines bring about the inflammation and try to attract only these cells. So into this environment, your Treg will not come because Treg does not like the inflammation pattern. It likes only it anti-inflammatory nature. So wherever there is no inflammation, there only Tregs can enter. So what the other cells do, if they go upper hand, what they do is they produce these cytokines, which brings about the inflammation so that this fellow will be completely shut down.
Treg cells can act upon the cells
By itself. Don't know, they have not shown such activity, but immunosuppressors, they act on other groups of cells. But this is another new view. Maybe they would have really worked, but I have no idea. But they have shown that they can act against dendritic cells, they can act against macrophages, they can act against T helper cells, T cytotoxic cells, even natural killer cells, neutrophils, all the other cells of immune system, whatever I mentioned. For all the other cells, this fellow can act and stop them. But it can, whether it can act by itself, I'm not sure.
Thank you.
But
You know, your immune cells will get suppressed if receptors are attached. Okay? So, who are the people we can apply T cell receptor? Only the patients or like he asked for normal people also we can only for the cancer patients or only for the
autoimmune conditions. Only for autoimmune diseases or transplantation stages only. Only those people lack this T regulatory cells. That is why they get this disease. So otherwise, the normal people are having bystander mechanism is we have the immunosuppressors, we have the T regulatory cells.
In the starting of the disease or before நான் கேட்கறது வந்து டிசீஸை வந்து கண்டுபிடிக்கிற முன்னாடியே அதாவது ஸ்டார்டிங் ஸ்டேஜ் இனிஷியல் ஸ்டேஜேவா மாதிரி
முதல்லயே கண்டுபிடிக்கலாமா யூ வாண்டட் டு சே
லைக் பேஷண்ட்க்கு வந்துட்டு ரொம்ப ஸ்டார்டிங் ஸ்டேஜ்லயே டைப் 1 அந்த மாதிரி அந்த ஸ்டேஜ்லயே நான் அப்படிதான் ரொம்ப யூஸ்ஃபுல்லா இருக்கும் ஆனா நீங்க சொல்றது வந்துட்டு
இல்ல அதுக்கு தான் what we can do is every month once in 3 months if you do a checkup no just a blood profile you can just check your T cell profile so there itself they will tell how much of your T helper cell is present, how much of T cytotoxic cell is present and how much T regulatory cell. So each has got his the basal value. So other which you can say whether if he or she has a lower value then you can say maybe you are having a lesser value so you may in due course you may get autoimmune condition. Other we can do. Yeah. Blood profile checkup nowadays people are checking even the you need the T lymphocyte population so the entire complete T lymphocyte population can be checked.
இது வந்து full body checkup பண்றாங்களா
blood checkup it's a WBC no so from WBC count you can check it up.
So it is better if we set up like 3 months.
Once in 3 months, how your blood? Because once in 3 months your blood gets replenished, no? Bone marrow is replenishing once in. That's why they say 3 months you can donate your blood and they are telling no. That's because every 3 months your blood is getting replenished.
Full body they generally in Tamil Nadu they are recommending only for 1 year. யாருமே 3 months once blood check-up பண்ணாதானே
Because they don't have time or anything. Yeah, once in a year.
மெட்ரோபாலிஸ் மாதிரி நேம் சொல்ல பட் எக்ஸாம்பிள் சொல்றேன். பிளட் சென்டர்ஸ் அவங்க ரெக்கமெண்ட் பண்றது
சரி, ஓகே.
மேபி ரெக்கமெண்ட் பண்ணா பேஷண்ட்ஸ் வந்துட்டு காசு ரெக்கமெண்ட் பண்றாங்க மேபி
They can feel, yeah.
அண்ட் கவர்மெண்ட்டும் டைம் இல்லாதனால
Okay.
There are no other questions. We can wind up now here. Okay. So let me thank. It was a really wonderful lecture today. Like insightful content and powerful delivery.
Yes.
So a big round of applause. Yeah, with that, I'd like to thank Anna Singh Library for as usual. Yeah, let's provide the space and
Thank you. Thank you.
this session. Thanks once again. Thank you, ma'am. Thanks everyone for attending this session. I think we have announcement.
So please give a feedback that the QR code is available.